And our last speaker in this session is going to be Etay from the Center for Addiction and Mental Health, CAMH, here in Toronto. And the talk is going to talk about modeling in human cortical microcircuits in mental disorders and aging.
Hello, everyone. So my lab does computational modeling of human cortical microcircuits, but I'm going to emphasize about the applications we have in mental health and aging. When we started about 7 years ago, we didn't think we'll get to it, really, but we are getting to it. An overview is I'm going to talk about the models themselves that we generated by integrating a lot of the kind of human data that you heard about today. And three main applications. One is trying to link mechanisms of disorders like depression, schizophrenia, aging to what the effect is on the cortical activity and function. The other theme is trying to, because we have detailed models, to simulate the EEG and then link it to EEG biomarkers that can be informative to in the clinic. And the last thing is we did quite a bit of work testing new pharmacology that targets microcircuit mechanisms. So our models integrate a variety of data. We were very excited to see that in the past 20 years there has been an accumulation of data. A lot of it is shared nicely by the Allen brain. Thank you. And there is also a lot from other groups in the literature and also from collaborators here at UHN. So when we started, there wasn't really a model of a human, detailed model of a human cortical microcircuit. So our first task was to generate one. So we developed models of the neuronal firing of the key cell types. We don't have 200 or 3,000. We limit ourselves to a humble 4. But those 4 are very distinct classes that are very important. There is the pyramidal, PV, SST, and VAP. All our models are constrained by human morphologies, human electrophysiology. Also, we're happy to see a lot of papers from some of the groups that have been talking today. Along the years, they've characterized pair recordings that, you know, now it's becoming more and more common, and so that's great. But back then, when we started in 2019, there wasn't still a lot of works, but there have been enough to constrain connections between, here you see a simulated pyramidal to SST and SST to pyramidal connections. It's a disynaptic inhibition. It's very important to maintaining the kind of excitability in the cortex. So we reproduced all of this in simulations. We also reproduced connectivity, which are the probabilities, and cell proportions, also from expression studies. So once we had those models, we could start asking the main questions that we set out to ask. If we have a candidate mechanism, for example, on the left, post-mortem data in depression patients show that there is a reduced SST expression in SST interneurons. So how do we determine the effects of this? People that have done those post-mortem studies from the hospital where we work, it's a mental health research hospital as well, have a lot of theories, and they do studies in rodents, but it remained as theories until you could link those changes to things you can measure, like impaired function and other things. So we simulated depression microcircuits with reduced SST. And in the middle, you see here, what we showed is that the reduced inhibition from SST specifically increased the baseline activity. So it essentially increases the noise in the circuit. Those cells are less active in the immediate response, the first part of the response. So the net effect is a reduced signal-to-noise ratio. And that essentially confirmed a lot of the hypotheses that people in the depression research community had. We took it a step further and tried to see, okay, what are the functional implications of it? Because SNR is a very kind of general measure. So what we did is we read out the output from the circuit. So if we look here in the distribution, the dotted line is the distribution of the baseline firing rates, and the solid line is the distribution of the response firing rate. What happens when you increase the noise, you increase the overlap between those two distributions. And if you were to tell whether, just by judging from cortical activity, if there was a signal or not, it's becoming harder because there is more overlap. And that overlap between the two curves is the failed and false detections at the signal detection task. And what we were able to show is that in depression microcircuit models, there is 2 to 3-fold increased detection errors. And nicely, you can see it in patients as well. So if you give them a very barely perceptible task to stimulus to detect, they are misdetecting it in about 2 to 3 times more than control. So that was a nice first work to try to link disease mechanisms to functional implications. The next thing we did more recently is we expanded those models to include the dendritic properties that have been characterized by Albert Gideon is in the crowd here, Gideon et al. From Larcom's group. There are not a lot of data on human dendritic properties, as most of you might know, but that was a good target to try to add to our models. So we modeled that. You can see the experimental curve in solid curve on the left. And the solid circles, the field circles, are the model that has this active backpropagation. But if you look at the passive model, the open circles, there is obviously a more dampened amplitude. So that's a demonstration of that. That work also added an important mechanism of depression, which is spine loss, which obviously affects the connectivity, but also the membrane surface area. And we were trying to see, okay, what are the effects on those kind of measures that we characterized before in the previous study. The first thing that we noticed, in red here, you see the backpropagating action potential at the apical dendrites. It's too high, and it shuts down too quickly. Whereas normally it would be moderate, and it will be smeared. And the implication of this is that if you activate the soma and then a little bit after activate the apical dendrite, so you trigger a spike at the soma and then it summates with incoming inputs in the apical dendrites, the black trace shows you the boosting effect that the backpropagating action potential has in healthy case. But in depression, because of this really shutdown of the apical action potential, there is no boosting. And if you see the curves, the black curves there, I don't know if this, is there a pointer here? Or did I press it? Maybe this. I'll try. So if you look at, on the left, the black trace versus the red trace, there is a dampening of the boosting effect that the backpropagation action potential has, and a loss of the nonlinear jump that you see here that you have in healthy. So that's one effect in single neurons. When we simulate the circuit on the right, we see the same thing. So when we activate the circuit, the recurrent activity dampens as a result. Is it stuck? Here. Is there a battery? Or? Delayed reaction. Right, so the next team of projects in my lab capitalizes on the detail of the models we have to simulate the resulting EEG. And I never thought that I would be analyzing and simulating EEG before, but turns out that for clinically relevant applications, it's pretty useful. It has a good temporal resolution. It shows you signatures of the dynamics of the circuit. and it's cost effective. So if MRI costs $1 million, let's say, then EEG doesn't cost much, right? So it has become pretty much one of our main applications, and therefore we set to simulate it. There are a few really great groups that develop tools to go from those neuronal microcircuit simulations to signals. One of the tools, the tool we use is LFPI. And the first thing we saw that was very exciting for us is that even though we didn't constrain anything about the EEG, our circuit generated a peak at the alpha frequencies, which are around 10 hertz, and they had a 1/f relationship between frequency and power. You see that on the left. So that was a nice validation that our models capture dynamics of the cortical microcircuits. So because we were able to trust them to replicate a healthy EEG, we were then looking to see what are the signatures of those cellular changes in SST inhibition. And the importance of this is that if somebody that has depression comes to the clinic, you don't know what the mechanism is. Even if you can measure their EEG, it could be any kind of mechanism. So you don't know what maybe treatment could be useful for them, and you don't know much. It's a very much of a black box situation. But going from the detailed models to simulated EEG gives us a link. And what we were able to see is a broadband increase in power, and that's in line with some cases of treatment-resistant depression. So there are definitely clinical studies that show those changes. So more recently, and we just submitted a paper about it, we took those in silico biomarkers, as we call them, and we applied them on real patient data to try to stratify subjects to ones that do exhibit those biomarkers. So the approach we take on the left is the first step. We train ANN or any kind of machine learning on the in silico data. That ANN learns from EEG features like the peak in a certain frequency band or the broadband parameters to infer what is the underlying level of inhibition from SST. And once we have that trained, and we get them to train fairly well, we apply it on patient data. And it's pretty much the beginning, but we've already, I think in 2 years, got to a point that we learned a lot from doing that. A few nice things that we found is that, first of all, we're able to find patients that exhibit those in silico biomarkers, because easily it could have been that we really don't see any patients showing that. But about a quarter of them do, and from the post-mortem data that I mentioned before, where you measure SST expression, they've seen it in about 40 to 50% of the patients, so we're looking at the same order of magnitude, so that's nice. Reminding you also that a change in expression is not necessarily going to lead to a change in EEG. So that was one nice confirmation that we're on a track here. So here on the top right, you see the group, we call them SSTR, that show a reduction biomarkers compared to the healthy and compared to the ones that don't in terms of their estimated inhibition. We quantify it in Z-score, so there are on average more than 2 standard deviations from the healthy. At the bottom, what was interesting is that the SSTR patients, when you look at their EEG, they're very different from the healthy. The SSTN, the ones that don't exhibit the biomarkers, are not that different. And there is way more work to do about it, but that's a pretty strong difference in terms of EEG. You can easily identify those people. But symptoms, we have not yet found distinguishing symptoms of those SSTR, which really demonstrate the utility of EEG. Because normally, how do you diagnose a person? You ask them, how are you feeling? How did you sleep last night? There's a lot of questions. There are some scores of certain tasks. But we are hoping that this demonstrates that EEG can be a pretty powerful signal to help diagnose. So yeah. The last team of projects in our lab simulates pharmacology for depression. So we have collaborators at CAMH, that's the mental health center that I work in that developed pharmacology that specifically boosts this SST inhibition back to normal. They only tried it on rodents. Those receptors, alpha-5 GABA, are the ones that SST target. So the drug is called alpha-5 PAM. It's a positive allosteric modulator, so it boosts the efficacy of those receptors. So in this project, we collaborated with Taufik, and they and the people that developed this pharmacology, they applied it to human slices to see the effect of that pharmacology on the pyramidal human neurons. We modeled those effects. Here on the top right, you see the measured and modeled currents with GABA alone and also GABA plus this drug. You see that there is an increase of the inhibitory currents because of the drug. Our models enabled us to estimate what is the effect in the dendrites, which obviously we don't know experimentally. More importantly, we were able to then test this pharmacology in silico on circuits, something that we can't readily do right now. Clinical trials are $2 million away and probably a couple of years away. So we started with that, and we were very happy to see that the dose that shows efficacy in rodents will do a good job in humans. You see the blue recovers the firing rate, baseline, and post-stimulus back to healthy levels. And also those metrics that I talked about before, the failed and false detection. So it suggests that it has a good effect on function. In the next study, we did things a bit more systematically. So we simulated a lot of microcircuits with different levels of reduced inhibition, and then tested, like simulated different doses to build a dose prediction function. I'm gonna zip through it because we've already delayed a few times. But the idea is that we find the dose or the bunch of doses that will get the EEG of a person back to the healthy levels. And when we do it across subjects, we can build a function. It could be an ANN or SVM or even linear regression that takes values from EEG and tells you what the dose you need to prescribe. Obviously, the implications for clinical trials are very useful because it can avoid just dosing people with unnecessary doses. And we show that that approach in silico recovers firing rates, function, and EEG back to the healthy level. The healthy level here for EEG is that cube, the gray cube. I'll quickly mention the more new and very exciting application. We use sort of the similar approach in aging. So that study, we looked for all the key microcircuit mechanisms that we could get a hand on that are reliable, they're in humans. The key ones that we saw are loss of inhibitory neurons, spine loss, and NMDA loss. And we simulated them to have like middle-aged microcircuits and older microcircuit and try to see what are the effects of those mechanisms. So the first thing we saw is that those mechanisms dampen both baseline, but more importantly, recurrent activity. You can see here the red is pretty dampened. Those are old microcircuits. That's the recurrent activity from younger ones. So that's one effect. And we can find the implications on it when we try to, again, read out activity and classify was there a signal or not. There is a decrease about maybe 15% in the accuracy, which you also see in older people, like real people as well. I think a bit more strong finding in this study is that when we simulate EEG from the middle-aged and older microcircuits, we're able to actually reproduce a lot of the biomarkers, the changes that you see in older people for real. So like, even though we just took a few microcircuit mechanisms, and we'll definitely own it that there are probably many other aging mechanisms on the macro scale level and everything, they were able to account for a lot of the kind of EEG property changes that, as I said before, are indicative of cortical dynamics. Maybe that makes sense because that very fast timescales are the timescales that maybe microcircuits are relevant for. But you can see the change. The experimental is the dashed line, and you see the simulated young and old. And the changes are pretty much in agreement with what you would expect. And I'll just finish this part by saying that the next part, like we did for the depression, is to go for real data. So we went to the Lemon aging data set, and we tried to essentially estimate the microcircuit aging from a person EEG. And again, there is no way you can do it in living people unless you kill them and do postmortem, right? Because that's the only way we get any kind of cell data from people. So like EEG and then estimating those mechanisms is sort of the first step to gauging the microcircuit components from a living person. The nice thing is obviously we see some kind of, I mean, it's not going to be one-to-one, but there is, older people do tend to be microcircuit aging, like older. And there is a nice moderate correlation with quite a bunch of cognitive metrics and impairments that are related to aging. So yeah, to summarize, I showed you our detailed models of human cortical microcircuits. I'll emphasize that our idea of why we're even doing it, and this crowd probably doesn't need too much convincing, but the main thing is we feel like we have a better prediction power for human aging and disorders. It's nice that there are some preclinical animal models for almost anything, but at the end of the day, schizophrenia in people is probably going to be a bit different, and depression in people is going to be. So when we study human disorders, we are happy to have human models. I showed you that we can use those models to link putative mechanisms of the kind that come from expression, genetics, and everything to cortical activity, function, and EEG biomarkers, something that currently experimentally is pretty tough to do. Maybe impossible, but I don't want to say that. And we showed you two very strong applications that we pretty much are very heavily working on these years. One is to estimate mechanisms from EEG, so go back, and then that can improve diagnosis. And also, like I said, if you have this pharmacology that targets SST, you want to know from all the, like candidate people who might benefit from this pharmacology. So if you have biomarkers that can suggest it, you're not going to give wrong medication for people. You're going to be more precise. And related to that testing, the pharmacology really helps translation efforts. And we're hoping that in the next year or so, it will be serving a very clear role for dose prediction in clinical trials. So yeah, I want to thank my great trainees. Alex is a postdoc, did a lot of the aging and testing pharmacology. Kent does depression and developing the models. And Frank did a lot of the EEG work. And thank you for listening.
Thank you very much for this.
Thanks a lot for your talk. In your microcircuit, you had to make some hypothesis, say, of whatever, the background synaptic activity or something that you cannot measure. What would you say to experimentalists that needs to be measured? What would you need?
I would love in vivo activity in humans and relating it to, I mean, there is some, by the way, but just not, when you have surgery that you can get some of this information, but a lot of let's say we constrain the response of the network, a lot of it comes from rodent papers rather than humans, although there is great neuropixels recent data, and Kostas, I think, mentioned some of that, and so we're looking to try to get that. I think a lot of the building blocks are nicely there, like we have electrophysiology. I would say also a region-specific, and I think there was a lot of questions about it today. We want to get some data from regions that are relevant for those disorders. So MTG is nice, but like PFC would be more relevant. Sometimes the visual cortex is even relevant to some conditions, so I think maybe guiding the efforts in that direction, just to name a few. I have a longer list, but yeah.
Hi, great talk. Concerning the alpha-5 data that you showed, can I see this slide again? There was something I thought was very interesting.
If I can get to it. Stop me when you want. This
Here,
one first?
yeah. So two things are happening here. You clearly boost the activity of GABA A receptors, but you see two effects. One is the amplitude of the synaptic events being higher, and the other is this tonic GABA current.
Yeah.
Super interesting. Do you think with your methods you could actually separate what is the therapeutic effect here? Is it the tonic current, is it synaptic current, or do you need both?
We should have done it, and yes, we can. I don't think I don't remember if we checked it quickly at any point, but it's definitely a good question. I think that the effect is there, though. So, like, I mean, we were talking about it. Maybe it can guide, because that group keeps developing variations of the drug. If there is a variation that affects maybe one type or not the other, as far as I know, there is some dependency. So I'm not sure you can decouple them completely, but that's a good question. We can do it in simulations and guide some experiments.
Because then you could also design more specific drugs, right?
Yeah, exactly.
Thank you.
Yeah, I wanted to ask about the deconvolution of the EEG patterns and relating it to the circuit motifs. One of the things that circuit does is respond very acutely to neuromodulatory tones. So, for example, cholinergic receptors on the VIP interneurons will kick in the whole SST circuit. In any of the EEG recordings, are there measures from trials when they're given oddball stimuli or sensory stimuli which would relate to a neuromodulatory change, which might be indicative of that SST differential?
Yeah, it's a good question. First of all, we're very interested to look at neuromodulation, and there is definitely modulation on that. But we, in a separate work that I didn't show here, we studied more schizophrenia-related questions, where the oddball is a key biomarker, and we were able to link it to PV and SST loss. So yeah, definitely that same approach we've applied. Yeah.
Super cool.
Mm-hmm.
That seemed like a really great demonstration of the use of the model and the simulated EEG, and really great that you've got this alpha-5 PAM. Just wondering if you know anything yet about the safety profile of that drug?
I think it's fine, but first of all, I'll remind again, in rodents, it's great. It's a more specific one, and I didn't show it here. I had to be selective. I had a lot of figures that I just cleared at some point, and I was exactly on time anyway. But benzodiazepine, which just affects inhibition everywhere because it affects the alpha-1 as well. It's like pretty much all the inhibition you see it would affect. Is very crude and doesn't improve cognition. It affects mood, but with a lot of side effects and also not good enough to recover things. So that drug being more targeted is somewhat safer, right? I'm not entirely sure if they finished all their safety measures, but yeah, that's exactly the phase essentially. But it's promising. It has a lot of therapeutic effect. It grows dendrites because chronic depression actually leads to dendritic loss, and that drug boosts neurotrophic effects that grow them back. So there are more indication that it's doing a lot of good than not. So yeah.
Thank you very much again for all the 4 speakers, and we will have a coffee break and be back in 3:15.